Hello, everyone, and welcome to New Frontiers in Gut Research. I'm your host, Paruni Krishnan, and I'm a third-year undergraduate student in the Honors Biology program at McMaster University. I am also an outreach coordinator in McMaster's Synthetic Biology research team.
Hello, everyone. My name is Pericles Vasiliou, and I'll be your co-host for today. I'm also a third-year undergraduate student at the University of Thessaly in the Department of Biochemistry and Biotechnology, and I'm currently working as a wet lab member in the Synthetic Biology research team iGEM Thessaly 2021. So, joining us today are two acclaimed researchers that will talk about their respective fields regarding the human gut microbiome and its effect on our well-being.
We hope that by the end of today's discussion, you are inspired to pursue the field of gut research. Our first guest today is Dr. Brian Coombs, who is a professor and department chair at McMaster University's Department of Biochemistry and Biomedical Sciences, and the Canada Research Chair in Infectious Disease Pathogenesis. Please welcome Dr. Brian Coombs.
Nice to be here. Thanks for having me.
It's a pleasure having you. So, Dr. Coombs, you're one of the most renowned researchers in McMaster. Can you tell us how you got involved in the field of gastrointestinal research and what drew you to it?
Sure. I've been very interested in bacteria for a long time. I did my PhD in microbiology, and for my postdoctoral fellowship, moved to the University of British Columbia across the country, and started studying bacterial pathogens which cause, typically associated with sort of food poisoning, acute infectious gastroenteritis, for example, Salmonella typhimurium. And in the course of that postdoc, got really intrigued by not only how bacterial pathogens can overcome our body's natural defenses, but also how they manipulate and modify other bacteria within the GI tract once they colonize a human. I joined McMaster after my postdoc in 2006, now going back a long time. And when I started the group, I started also with Salmonella research, but also was interested in branching out a little bit into other pathogens. And we got quite interested in Crohn's disease, which is a chronic inflammatory disease in humans. And Crohn's disease is quite a fascinating disease because although the etiology of Crohn's is still complicated and not quite fully understood, in other words, the causes of Crohn's disease are not fully understood. What is generally accepted is that the composition of the microbiome in patients is a main driver. And so my group then got quite interested around 2008, 2009 to try and uncover bacteria which are associated with Crohn's disease so that we could better understand the disease process and think about new ways to treat it. So it's going back a long way, and it was sort of stemming from my postdoctoral research on bacterial pathogens.
So that's really interesting. And as you said, one big part of your research interest is Crohn's disease. So could you please tell our viewers a bit more about what you're currently working on?
Sure, yeah, happy to. So when we got into Crohn's disease research, going on 10, 12 years ago now, we were kind of coming at it from this microbiology angle. Me being a microbiologist and also a background in biochemistry, we were very interested to try and understand the microbes that are associated with Crohn's. And so the first projects and the first small grants that the group got just to enter into the Crohn's disease research field were to investigate microbes that are associated with Crohn's disease patients. And so we started out by taking biopsy samples of Crohn's patients and taking them back to the lab and culturing what was there, and following up on some work that was done and published back in the late 90s by a French group of Arlette Diffry-Michaud, who was also a gastroenterologist who was studying Crohn's patients and also trying to understand disease mechanisms from the microbiology angle. And what she found was, in the course of her research, was a unusual kind of E. coli that has now been known to become known as adherent invasive E. coli. And she described these adherent invasive E. coli as associated with Crohn's patients, highly enriched in Crohn's patients, but not so much in individuals without Crohn's disease. So it seemed to be sort of associated with the disease somehow. And these E. coli are quite unusual in that not only can they adhere to gut epithelial cells, but they can also invade into epithelial cells and other immune cells of our bodies where they can replicate and live. And our bodies have a very hard time trying to eradicate these particular kinds of bacteria. And so my group got very interested in AIEC. And at the time, there was almost nobody, there was a couple of groups in Canada working on AIC, but not really from the kind of perspective that we were trying to do, which was understand the disease mechanisms and its infection biology, sort of from a fundamental point of view. And at the time, I had a very talented postdoc in my group, Sherry Small, who also was quite interested in AIC and was quite a gifted animal experimentalist and also immunologist. And so we set out to establish a small animal model of AIC infection in mice, because at the time there was really no good animal model that we could use to advance our understanding of infection biology. And so through a lot of careful and quite exciting work actually that was going on in the group at the time, Sherry developed this very nice mouse model of AIC infection, which allowed us to monitor how AIC infects animals over chronic timescales. So it was the first animal model of AIC, which was that we published in 2013, that allowed us to essentially colonize mice for life and understand how AIC changes the immune response in those animals and also the microbiota within those animals. And it's been quite a useful model for us. We've advanced it several times and expanded on it and made it better. And also we've helped private sector companies who are interested in developing new Crohn's medicines to implement these models within their organizations to help advance drug discovery for Crohn's disease as well. So it's been quite an interesting and fun run in the lab, and we have a lot more things going on in the lab with AIC that we're quite excited about as well.
That's incredibly fascinating. I know you briefly mentioned the etiology of Crohn's disease. So my follow-up to that would be, is Crohn's disease caused by genetic factors, environmental factors, or even both? And also, what are some risk factors associated with Crohn's disease?
Yeah, I mean, in terms of causality, I mean, it's kind of all of the above that you mentioned. Genetics certainly plays a role, because in first-degree relatives, among first-degree relatives, Crohn's disease is more likely to occur. Know that there are genetic predispositions to Crohn's disease. So people have been studying the genetics of Crohn's for a long time. And we now know of probably about 200 mutations within different genes, targeting barrier function in the GI tract, immune response, and other things that increase your risk for developing Crohn's at some point in life. However, any one genetic risk factor only accounts for a very small percentage of risk. And so we do know that there's things beyond genetics which play a role. People have looked to the so-called environment, which is the catch-all bin to put everything that we don't quite understand what might be involved. But these are things like lifestyle factors such as diet, eating habits, smoking, stress, anxiety, things like that, depression. There's also medication, which we now know can be attributed to some degree of risk for Crohn's disease. For example, antibiotic use. So there's been some nice data out of the group in Canada, in Manitoba, looking at antibiotic use in adults and in children, and demonstrating quite significantly that high antibiotic use among children can increase their risk of Crohn's disease later in life, and various other medications as well. So it's, there's a lot of things, I think, which feed into Crohn's disease risk. we don't quite understand how all of these different risks interact with one another. You know, the way we think about it in the lab is that Crohn's disease is probably not caused by one thing. It's a constellation of different risks and that come together in an individual that predispose them to disease. And so the group is using our animal model as well to try and understand, you know, what we call this multi-hit hypothesis for Crohn's disease, meaning it's not one thing that it's going to happen to you that's going to lead you to develop Crohn's disease. It's probably a constellation of things that are, you know, primary insults, secondary insults, tertiary insults that over time wear down the homeostasis in the gut and predispose you to a disease state. And under the right circumstances, and we believe, you know, bacteria, the right constellation of bacteria in the gut at the time can also play a role. So that, I mean, that's the risk stuff. There's lots of known risk factors which feed into Crohn's disease. Is any one of them, you know, required for the disease? Probably not. It's probably a mixture of different disease risk factors coming together in an individual based on genetic predisposition that lead to disease. And second part of your question, I forget the second question you had.
Oh, it was just what were the risk factors? So
Oh, okay.
Yes.
Yeah. Okay. Yeah. So I mean, the things that we've been talking about, you know, things like antibiotic use. We know also that, you know, episodes of food poisoning, so acute infectious gastroenteritis caused by enteric pathogens like Salmonella, like pathogenic E. coli, like Helicobacter jejuni. We know that those also increase one's risk for Crohn's disease over a fairly long time scale. So there's been some really interesting and beautiful population-based data out of mainly Europe, because these studies are quite large, because they have you know, more careful data health database records. So these, you know, these trials have tens of thousands of individuals in them, and it is quite significant that if you have one or more episodes of acute infectious gastroenteritis, your risk over 10 years of developing Crohn's disease after that first acute infectious event is significantly higher than if you weren't exposed to the infectious agent in the first place. And that's you know, it's quite interesting and curious, because, you know, for those in the audience who have ever had acute infectious gastroenteritis, it's very unpleasant, but it doesn't last forever. I mean, your body is pretty good at, you know, controlling most of the bacteria which cause self-limiting gastroenteritis, like Salmonella, pathogenic E. coli, Yersinia, Campylobacter. You know, at most you might feel really poor for about a week or more, 10 days, 2 weeks, something like that. But then your body recovers, and you do mount a pretty good immune response, and you eradicate the pathogen. So it's quite interesting then that why is it that after the pathogen is gone, your risk of developing Crohn's disease persists for, you know, over a decade after that first event. The way we think about it in the lab is that probably that acute infectious gastroenteritis event creates, again, a disturbed balance in the gut because of the vigorous inflammation that gets generated. Perhaps there's new microbes which can come in, new bacteria which can, you know, establish themselves in the GI tract after those periods of time, which then persist and increase your risk of Crohn's disease. That's one hypothesis that we have studied in the lab, actually, and published some work on. But there's probably other things involved too that increase your risk long-term after the pathogen is gone.
So as far as gastrointestinal diseases go, and the whole complexity thing for all of the risk factors that contribute to these diseases, we all can certainly think that gut microbiome plays an important role in developing these diseases. So how do you think that the composition of the gut bacteria relate to conditions involving chronic inflammation?
Yeah, it's a great question. So, you know, there's been a lot of published work on, and this isn't work that my group's done, but, you know, there's a lot of groups around the world who are looking at microbiome composition in Crohn's patients. And there's been some, there's a lot of papers published now on this. And again, when you get down into the granular details of, which bacterial species are there, it's sometimes hard to compare from study to study because the study designs are a little bit different. But there are some common themes. And in the context of the things that my group works on, one of the common observations in Crohn's patients is that they tend to have an enrichment or an overabundance of Enterobacteriaceae. And when you drill down a little bit deeper about which Enterobacteriaceae are there, oftentimes you find, adherent invasive E. coli. And the interesting thing is that with AIC, because they are adherent and because they can invade into tissues, it doesn't mean that they're limited to the lumen of the GI tract. So they can live in the lumen of the GI tract perfectly fine. And we know this from humans and also in the animal model that we've been working on. But quite quickly after colonization, the AIC population sort of diverges and bifurcates into two areas. So you get a population which remains in the lumen, and that population of bacteria can compete very well with other members of the microbiota, where there's a very high density of bacteria. But because they can attach and form biofilms on the surface of gut epithelial cells and invade into epithelial cells, they can gain access to what we call privileged niches in the GI tract. So places where we don't expect or want or bacteria to be, which is in the lamina propria. Our bodies are exquisitely engineered to maintain bacteria in the lumen of the GI tract and prevent them from gaining access to any other site of the body. And so because AICs are adapted to invade and evade our immune system, they gain access to these sites within our bodies, which when bacteria are there, our immune system mounts a very vigorous immune response to them, creating inflammation. And so we think part of the reason why inflammation is chronic in Crohn's patients is because probably there are bacteria that can gain access to privileged places in the body where our immune response is programmed to respond to. Our immune response is pretty good at forgetting and ignoring the vast number of bacteria that are in the lumen of the GI tract, but they're exquisitely primed to deal with bacteria where they escaped that containment and invade into deeper tissues of the body. And so we think that bacteria like adherent invasive E. coli could be one of the main drivers because they can chronically stimulate the immune system over and over and over again from this luminal population. So that's one of the things that we've been studying, and also to then trying to understand, well, Enterobacteriaceae are expanded in Crohn's disease. What happens if you try and reduce the population of Enterobacteriaceae? So you get these big Enterobacteriaceae blooms. What happens if you decrease the size of the Enterobacteriaceae bloom or prevent them from blooming in the first place? Does that change the disease outcome or the disease course? Does it help resolve disease for some patients? So this is an open question that is not resolved at all in certainly in humans. We're starting to begin to understand this in our mouse model.
That's incredibly fascinating. And I know that the discussion was mainly surrounded on how pathogenic bacteria affect Crohn's disease. So now we were wondering, are there ways bacteria can be beneficial to us? And also, are there ways bacteria can be beneficial to patients with Crohn's disease?
Absolutely. I mean, the vast majority, 99% of the bacteria in our GI tract right now are helpful to us. And, we have evolved with them forever. And, we have this sort of very close symbiotic relationship with our microbes. And indeed, most of the bacteria in our GI tract are protective. They help us, harvest nutrients from our food. They generate vitamins and minerals for us, and they do all kinds of things. And one of the main beneficial things they do is provide a state which we call colonization resistance. And colonization resistance is, you know, as the name implies, it's a state where the bacteria that are colonizing our GI tract exclude and prevent foreign bacteria, which we don't want there, like pathogenic bacteria, from colonizing. And so they resist colonization by pathogenic microbes. And so, of course, the problem with antibiotics is they kill indiscriminately. You know, they kill harmful bacteria, of course, because that's why we need them. But in the process of killing harmful bacteria, we also kill and damage helpful bacteria. And it's well known in many, many models, and in humans too, that antibiotic use in some cases can increase your risk of developing secondary kinds of infection. A good example is Clostridium difficile. Chronic use of antibiotics can increase your risk of developing Clostridium difficile infections in your GI tract. You know, whether or not those are Clostridium difficile that come in new because of the antibiotic use and creates a niche for them, or in some cases they might be there already in very low numbers, but the commensal microbes in our GI tract keep them in check. And so when we damage them and knock back the numbers of helpful commensal microbes, we increase the opportunity of pathogenic ones to expand. I mean, so that's one example, and there's certainly other examples as well, and also in animal models. So yes, there's helpful bacteria, and you know, I think as we have started to understand more about how the microbiota is associated with health and disease, and not just for gut diseases, but all kinds of other conditions in our bodies. You know, to me, I think the next holy grail of microbiome research and also infection treatment research is try and figure out ways to more precisely target harmful bacteria so that we have, you know, I would say more limited spectrum antibiotics, because we need antibiotics. We're never going to not need antibiotics. We have to have them. They help, you know, they save lives. But we want to preserve healthy bacteria as much as we can. And so if we can come up with more precision therapeutic methods for harmful bacteria, I think that that's going to you know, revolutionize our ability to treat infections while at the same time preserving the healthy microbes in our GI tract, which help keep us healthy in the first place.
This is really interesting. And what I'd like to ask you now is if you could like point out some key features that separate commensal from pathogenic bacteria, and if there exists a species of bacteria that can act as both commensal or pathogenic.
Yeah, so I mean, there's, you know, maybe I'll answer your second question first, which is, you know, are there species which can be commensal and pathogenic? I mean, that's certainly true. You know, for example, all of us have Staph aureus on our skin right now. They're part of our commensal skin microbiome, but there's also very nasty Staph aureus type of bacteria like methicillin-resistant Staph aureus, which, you know, when it gets into, when it breaches your skin and can cause deeper tissue infections, is a serious problem to try and treat. So there are lots of examples of how bacteria exist on this spectrum of lifestyles, from commensal to pathogenic and then somewhere in between. I would say adherent invasive E. coli that we study in the context of Crohn's disease is probably on that spectrum as well. AICs are clearly derived from many different kinds of E. coli, which we know don't really cause infection, but they're also not as pathogenic as you know, things like enterohemorrhagic E. coli or enteropathogenic E. coli. So they're clearly not like, you know, exquisitely frank pathogens, but they're also not commensal either. They're somewhere in between. So maybe they're sort of on this spectrum of evolution. You know, when I think about what differentiates commensals from pathogens, probably where my head goes to is the pathogen's ability to cause damage to the host. Commensal microbes don't cause damage. They sort of live in harmony with us. Our immune systems are largely tolerant to them, and we don't mount vigorous immune responses to them. Pathogens, on the other hand, our bodies do treat as foreign. We do mount vigorous immune responses to them, and they cause extreme inflammation. And it's the inflammation typically which is causing damage to host tissue. So that's one key feature, I think. And probably the reason in some cases with some bacterial pathogens, why they cause inflammation and damage is because they can get into places of the body that our bodies typically like to exclude bacteria from. You know, the GI tract is an interesting example because, you know, we have all kinds of innate defenses in the GI tract, which are, you know, whose sole purpose is to keep bacteria in the lumen of the GI tract. You know, we have immune cell surveillance in the GI tract that prevents pathogens and samples pathogens constantly, sampling what bacteria are there and sort of sniffing out the pathogens, if you will. We've got cells which make antimicrobial peptides, so sort of our body's natural antibiotics, which prevent bacteria from getting close to the epithelial border. We've got other specialized cells which make thick mucus in the GI tract, which create a physical barrier, making it very difficult for bacteria to get access to the epithelial surface. And then you've got this single layer of epithelial cells which create this, you know, tremendous barrier to invasion by, you know, the literally trillions of bacteria which are on the other side. And it's the pathogens which have evolved ways to, you know, I guess you could, you know, they, in some cases they can root out the deficiencies in the border, in the barrier. So they can, you know, interfere with tight junctions, which keep the barrier intact. They can invade into the cells and just, you know, basically use epithelial cells as a route to get to the other side, the basolateral surface, for example. So they have evolved sophisticated ways to get around the barrier defenses into places where our bodies don't want them to be. And as soon as our immune system detects them in places we don't want them, that's when the immune response is invoked and inflammation ensues and host damage ensues. So I think of, you know, the distinction between pathogen and commensal as one of causing damage to the host, and the damage, you know, primarily because of, you know, location. To me, it all comes down to location. You know, our bodies have evolved ways to keep the bacteria that we need, like the commensals, compartmentalized into places where we can contain them and control them. And as soon as you get a bacteria in there that can, you know, sort of, you know, cut holes in the chain link fence and break through, then that's when our immune response kicks into high gear.
That's very interesting. And considering you mentioned the implications that the overuse of antibiotics has, and I'm sure our audience is familiar with the fact that antibiotics is used to treat Crohn's disease. So I was wondering, could you elaborate further on some alternative treatments that can be used to combat the issue of antibiotic resistance that's associated with the overuse of antibiotics?
So you mean how do we come up with a better antibiotic? Is that what your question is?
Or any other alternative forms of treatment for Crohn's disease?
Oh, for Crohn's disease. Okay, well, I mean, I'll tackle your first question about alternatives to antibiotics. I mean, there's lots of research going on in here, and you know, the alternatives to antibiotics research is largely fueled by the global crisis that we're in, which is antimicrobial resistance. You know, it's been called the next slow-moving pandemic. You know, we have bacterial infections now for which there is no pill on the shelf which can treat you. There are bacteria which are resistant to every single antibiotic in clinical use. And so, yes, there is a massive campaign on to find new antibiotics. So people are trying to find new conventional antibiotics that act in conventional ways, you know, hitting classical targets in bacteria, but that, you know, like DNA replication, RNA transcription, cell wall biosynthesis, you know, those ribosome biogenesis, those classical targets. And it's quite possible that new antibiotics will be found that hit those new targets and that are not susceptible to existing resistance mechanisms. But we also know that resistance is inevitable. And, you know, if you find a really potent antibiotic, you know, there is no antibiotic that's evolution proof, and there's no antibiotic that's resistance proof. The bacteria will find a way around it. So people are looking at alternatives. And, one of the things, so in group, we have, an entire arm of our research is trying to find alternatives to antibiotics. And one of the things that we're trying to do is leverage and harness the power of the immune system, the innate immune system. The innate immune system is fascinating, and in a way, collectively, all of the mechanisms of innate immunity are, in essence, the perfect antibiotic. We've got cells which are, making potent chemistry, which can damage and destroy foreign microbes. They make natural antibiotics like antimicrobial peptides. They make reactive oxygen species and reactive nitrogen species. They make all kinds of really interesting and important chemistry that damage bacteria. And it's really hard for bacteria to evade innate immunity because the innate immune system is hitting them from all different angles, whereas an antibiotic pill is hitting them from one angle only. So I guess my first answer is I think we have to hit bacteria from multiple sides. So people are looking at antibiotic combinations. So using combinations of drugs which hit bacteria from two different directions, making it more difficult for them to evolve resistance to. And we're leveraging that concept in the lab to try and understand, well, can we, if combination therapy is good, which we know it is, can we use conventional antibiotics in conjunction, or new chemical matter for that matter, in conjunction with some of these chemistries of the innate immune system? So we're trying to study small molecules and identify small molecules in high throughput screens, which increase and work together with the chemistry of the innate immune system synergistically to kill drug-resistant bacteria. So that's one area is combination therapy using harnessing the power of the immune system is one area. Phage therapy is also a thing which is coming back into laser beam focus right now. My group does not work on phage therapy, but there's plenty of other groups who are doing outstanding phage research. Phage are interesting, bacteriophage, because we've been talking about narrow spectrum precision antibiotics, but in a way, phage, that's exactly what they do. They hit, in some cases, they infect a single species, or in other cases, even more narrowly, different strains of a species of bacteria. So bacteriophage are really the nature's precision smart bomb. They can get into and find out their own specific bacteria that they want, host bacteria that they want to infect among probably thousands or millions of other bacteria which they don't infect. And so people are trying to leverage the power of phage now to not only overcome antibiotic resistance, but develop more smart precision-based therapeutics. So, there's lots of different ways people are trying to overcome the antibiotic resistance problem, and it will work. We will solve this problem. We have to. We have no choice. So there's tremendous resources being poured into antimicrobial resistance research. Shifting to Crohn's treatment, clearly there are unmet clinical needs in Crohn's disease treatment. The vast majority of Crohn's therapy right now target the inflammation. So they're immunosuppressive or immunomodulatory drugs which suppress different arms of the immune system to try and dampen down the chronic inflammation in the GI tract. The challenge with those kinds of therapies is, although they work, they don't work in everybody, because we don't, I think, fully understand inflammation in all Crohn's patients, because Crohn's disease is, I would say, a very heterogeneous disease. It's not the same thing in different people. So the nature of the inflammation can be different. So the current therapies don't work in everybody. And even in people where it does work initially, oftentimes those people over time will lose responsiveness to the therapy and it won't work anymore. So, one of the things that people are trying to understand is identifying the root cause of the inflammation. So, immunosuppressive drugs, they address the symptoms, they don't address the cause of the symptoms. And the analogy I give to people sometimes is like, if you're working on a tap in your house, and all of a sudden the handle on the tap breaks off and the water starts shooting out, you have there's two things you can do. One, you can put your hand on where the water's shooting out to stop the water from flooding your house. And that's kind of like the way I look at Crohn's disease treatments now. It's trying to shut off the inflammation, not really by dealing with the problem, but by sticking your hand over the faucet to prevent the water from shooting out. But as soon as you take your hand away, the water comes back. A more durable long-term solution would be to find a new tap, reattach it to the tap, or the new handle, reattach it to the tap, and turn the water off. And so we're, I guess, trying to understand where the handle is in Crohn's disease. We don't quite understand where the inflammation is coming from, but clearly the bacterial composition, is a smoking gun. We think the bacterial composition in Crohn's is playing a big role in the inflammation. And so there are companies now, lots of companies, who are trying to target what we think are disease-causing microbes in the GI tract using precision therapeutics to see if that has a role in preventing more inflammation. And these drugs would probably be used in combination and in conjunction with existing anti-inflammatory drugs, like biologic therapy that hit, TNF alpha, for example. So again, a dual targeting approach, hitting the disease from more than one angle. And right now, I would say the microbiome is the clinical unmet need in Crohn's disease. And there's lots of companies who are investing tremendous resources into figuring this out.
It is really interesting, actually, to hear where today's therapeutic research focuses on. So having that said, and based on your experience, I would like to ask you, what are your predictions for progress on treating inflammatory bowel diseases in the next decade?
Yeah, that's a good question. Certainly, as I've been alluding to, I think targeting the microbiome is going to be a major thing. To me, I think it's the next holy grail in Crohn's disease. We have a pretty good understanding of the inflammation. We have good drugs now to try and dampen the inflammation in Crohn's disease that works in a lot of people. But I would say that targeting the microbiome and editing the microbiome to a more healthy state is going to be sort of the next holy grail over the next 10 years. The other thing that I think is probably going to come is trying to develop more site-specific treatments for Crohn's disease. Right now, the really good biologic drugs, like the anti-TNF alpha monoclonal antibody therapy, is delivered systemically, and so you get whole body depletion, this major immune response, and it leads to other secondary complications like risk of infection and other things. And if you could target that response directly to the GI tract in some directed way, then you might have you'd still have the benefit of this really amazing therapeutic, but it's acting locally at the site where the inflammation is, and you're in a way preserving that immune response elsewhere in the body where it's needed. So I think that could also be another area for the next 10 years to think about is more locally acting site-specific treatments in Crohn's disease that are targeted locally to the GI tract.
It's very interesting to learn the importance of site-specific treatments, and there's a lot of potential I can see with that in the future. And also, for our last question now, what advice do you have for students that are planning to pursue research in the field of gastroenterology?
My advice is, it's a super exciting field. My general advice to anybody who's interested in doing research is to, jump in with both feet and, pick a topic that you're passionate about. When I was an undergrad student, I thought I was destined to go to medical school and become a clinician. And then in my third year, I worked in a lab in the summertime doing research. And, from that first week in the lab, I realized that probably medical school was not for me. I loved research. I loved working with my hands. I love thinking about problems. I love discovering things and, you know, seeing something for the first time that, you know, nobody else in the world has ever seen before or discovered before. It's really, to me, research is really exciting. And then when you pick a topic that is, you know, you're passionate about, it almost doesn't feel like a job anymore. I mean, it just feels like you're, you know, you're just playing all day long. I love that about research. And I, you know, I don't spend obviously time in doing research and working in the lab myself. The, you know, folks in my lab are doing those things now, but certainly it's still an opportunity to, you know, keep thinking about new ways to solve problems, working with the next generation of young people, young scientists who are also eager to and excited by research. I mean, it's truly a, for me, it's my dream job. So yeah, I think if you have an interest in research, jump in with both feet, pick a topic you're passionate about, and success will come.
All right. Thank you so much for joining us today, Dr. Coombs. It was a real pleasure having you here with us. So now we are going to be moving on to the audience Q&A portion. So please feel free to leave your questions in the chat below, and I will be reading them out in 30 seconds. So we will be back in 30 seconds. So welcome back, everyone. So I see here we have a question from Maya. So she's asking, how are mice adapted to replicate crumbs?
So it's a good question. You know, mice are typically the go-to small animal to study preclinical models before we get into humans. They're not a perfect replicate of humans, of course, but they do have certain features which make them useful. One is that they have, you know, an innate and adaptive system quite similar to ours. They have the same cells, immune cells as we do, and they also have a microbiome, and so we can study them within that context. So we can have mice which have you know, a normal mouse microbiome, which, you know, in fact has many of the same microbes as we do in our GI tract. But we can also modify and adapt these mice to, for example, have a human microbiome. So we can raise mice which have no microbes to begin with. So we call these azenic mice, so they're germ-free. We can put microbiome bacteria from patients, either Crohn's patients or healthy individuals, back into those mice and have those different microbiomes establish themselves in those mice. So they're quite, I guess, amenable to, manipulating both the microbiome composition and also the immune composition. So we can also study, for example, things like knockout mice, which are depleted in a certain component of the immune system, or transgenic mice, which express, a human variant of a disease-associated protein or something like that. So I guess they're useful models in the sense that they have, the same kind of immune cells and that we do, and also bacteria, which are similar to ours as well. And so we can study their immune response, and by doing that, of course, the work that we do in mice has to be replicated and validated in humans at some point in order to increase its clinical relevance. But oftentimes we will start in mice because of these beneficial, these things which make them useful for scientific research.
All right, thank you. We also have a question from Costas here. The question is, does colonization resistance prevent the action of probiotic strains?
That's a good question. I mean, my instinct is to say probably, yes. Probiotics, there's probiotic bacteria and there's also probiotic yeast. And for the most part, these probiotic bacteria and yeast don't typically colonize the GI tract long term. So they might colonize transiently for a very short period of time, but they don't necessarily colonize and establish themselves as a commensal member then of the microbiome. So my pardon the pun, my gut is telling me that probably the commensal microbes do exclude probiotic bacteria and yeast from the GI tract. But nevertheless, it allows them to, at least colonize or transit through the GI tract long enough to elicit their beneficial effects on intestinal health.
All right, and I guess our final question here is from Jessica. So she asks, how can we leverage innate immunity? Is this similar to CAR-T cell therapy, where specialized innate cells are developed outside the body, or are we exploiting the signaling molecules?
Yeah, I mean, there's different ways of thinking about this. And I mean, the way we're thinking of exploiting innate immunity is by trying to figure out if we can, going back to this idea of combination therapy, if we can find either antibiotics or new small molecules which don't necessarily kill bacteria, but weaken them in some way to make them more susceptible to components of innate immunity, then that's kind of the way we're thinking about it. So for example, some of the screens we have done in our high throughput screening lab here, one of the components of innate immunity, for example, is antimicrobial peptides. So antimicrobial peptides are cationic peptides that some cells of our body make. They can interact with bacterial membranes and cause damage to those membranes and poke holes in them. Bacteria can become resistant to antimicrobial peptides because, through many different ways, but in one way, one mechanism is that they express a protease on their surface, which can cut these antimicrobial peptides at the dibasic sites, rendering them ineffective. In some cases, bacteria fortify the outer membrane, making it more difficult for antimicrobial peptides to gain access to the membrane surface of the bacteria. And so again, if you found a small molecule which either inhibited the protease on the surface or inhibited the enzymatic modifications to the bacterial outer membrane, preventing its fortification, now you would have a combination which works really well. You've got the antimicrobial peptide, which is great at killing bacteria because it can interact with the membranes. And you've got this secondary molecule which can inhibit the resistance mechanisms to the antimicrobial peptides. So the way we're thinking about leveraging innate immunity is by combination therapy, harnessing the chemistry of innate immunity as the secondary treatment in conjunction with novel small molecules. Hope that made sense.
And I think we have one final question here. So this is from Andrew, and he is asking if you're familiar with the gut to brain hypothesis and the impact of gut bacteria to neurodegenerative diseases.
Yes, I mean, this is one of the things that has come out of microbiome research over the last decade. Is it seems like there's no human disease that the biome doesn't influence. It seems to be way more pervasive than just sort of living in our GI tract. They influence all kinds of human health and disease conditions. So yeah, there is a huge body of research going on related to this, to Andrew's question around the gut-brain axis, and that is, how does microbes in the GI tract influence behavior? How does it influence from a long distance, how different parts of the body react to different stimuli, react to different drugs, react to different insults? Lots of research going on in this area. It's super fascinating. And again, this is probably over the next 10 years, we're gonna uncover even more mechanisms of how gut bacteria influence all kinds of things, including probably neurodegenerative diseases.
That's really fascinating. And I suppose that is all the time we have for the audience Q&A. Thank you again so much for joining us today, Dr. Coombs.
Thanks very much for having me. This was fun, and thanks for your interest in this topic. And yeah, keep going.
Thank you.
Thank you, Dr. Coombs. It was really nice having you here. So, moving on with our next guest, would like to welcome Dr. Alexandra Mazziti. Hello.
Hello, it's nice.
So thank you for joining us as well. So Dr. Mazziti is a postdoctoral researcher in the Department of Nutritional Sciences and Dietetics of International Hellenic University in Greece. So we know your research is centered around microbial ecology in aquatic environments, gut microbiome under changing conditions, and the human microbiome. But can you tell us a bit more about what you're currently working on?
Okay, first of all, thank you for inviting me. So all these things you said are true. I have worked with many things, but always I'm working with microbial ecology. Currently at the International Hellenic University, I'm trying to study the gut microbiome changes in different polyps, gut polyp samples. And we're trying to see how the microbiome changes, and we are trying to see if there is any relationship, correlation of the gut microbiome changes, depending on whether the patients have Helicobacter pylori infection or not. I could say that this is quite challenging because we are not working with feces that usually are used in gut microbiome studies, but we are working with tissue samples, like samples from biopsies. And we are trying to see whether these polyps are, how dangerous they are. So we also take the other data from the doctors. And we want to see if there are specific species, along with Helicobacter pylori, that make a change in order to characterize the polyp, if it is adenomatous or if it has the potential to be cancerous and other factors.
That's very interesting. And just as a sort of background question, I was wondering, what got you involved in the field of nutritional sciences, and also what drew you to it?
Okay, first of all, I have to tell you that I'm not hearing you very well. It's very clear, but it's not very loud, and I cannot put it louder, but I can hear most of it. Okay, so everything started when I was doing my bachelor thesis in 2004 in the University of Athens biology department. And I was always fascinated with sea biology. But I also liked, okay, at that time, all the molecular techniques were really emerging. So I wanted to find a way to combine these two. And the subject of my bachelor thesis was to study the gut microbiome of sea urchins and ascidians in order to find find different species and detect some potential compounds that could be used for biotechnological purposes. So this is how everything started. And after that, so this is how I entered in gut microbiology with some very simple animals such as sea urchins and marine ascidians. Since I started working with it during my thesis, I found it very fascinating. But then I found fascinating all the field of marine microbiology and marine microbial ecology. And I worked in several subjects through the years during my master's and during my PhD, but everything had to do with marine microbial ecology. And especially during my PhD, I worked also with gut microbiology, this time for prawns. Probably you have heard of there are not, you don't have the species in Canada. It's mostly European. And I was working, I wanted to check whether we have differences in natural populations that are collected from the field, and also they depend on a yearly basis. So do we have differences in the gut microbiology in different seasons and different months, and which are the reasons for this? Is it the different food that they can find? And then to find the differences in cultured populations of the prawns.
That's really interesting. So what I'd like to ask you next is that, so it is a well-known fact that our overall well-being is strongly dependent on the health status of the gut. Would you suggest from an ichthyological aspect that there is any evidence such a relationship could exist among fish as well?
Yes, of course, because we have worked with many marine, with some marine animals like fishes. And we know so far that, not only from us, but from many scientists, that the gut microbiome and differences in the gut microbiome are really important in order to have a good health in the fishes. So everybody's trying, even when they try different probiotics, the first thing is to see if this probiotic is a pathogen for the host. And of course, other scientists have also found that differences in the gut microbiome are really crucial for the host well-being. Like we know so far that growth rates in aquaculture are really influenced by the gut microbiome, that there are specific bacteria that really enhance good growth, the growth of the host. And there are other microbes that are detected in the gut microbiome that are really do not allow the host to grow as it is supposed to.
So this is really interesting. And besides a well-balanced diet, how do you think environmental factors such as climate change affect the biodiversity of microbial organisms, either in humans or animals?
Yes. So we are trying to start, we have applied for a big project. In order, we know so far that global change, especially for the Mediterranean, apart from temperature, as it would be expected, it also influences salinity. So we know that a very well-known fish, a very popular fish here in Greece and in other Mediterranean countries, which is the sea bream, can grow in different salinities. But we don't know how its growth is being affected by these differences in salinity. And also, we don't know so far, and this is what we want to check, and we hope we will be able to do it, how the gut microbiome will change in different salinities. Because, of course, in different salinities, different microbes thrive. And this is about the fishes and the marine animals, of course. And about humans, I think you also asked for humans, right?
Yeah, right.
Okay. So, of course, the changes in the environment also can affect the human microbiome, the gut human microbiome, because we also know that some studies have also shown that the effect of differences in microbes in the tap water that we consume are also affecting our gut microbiome. So potential changes in the quality and in the microbes that are present in the tap water that most of us consume will also be important for our gut microbes and how these gut microbial communities change.
That's very interesting. And considering our previous discussion surrounding antibiotics in the gut microbiome, I was wondering, can an individual's antibiotic overuse have a direct impact on the health of another individual?
If you take it, you see it so strict. I don't believe that. Like that, one individual overconsumes antibiotics and then another one would be affected. But in a broader view, I think that, of course, this is important because the overuse of antibiotics, we all know that it has already led to this resistance in antibiotics of most microbes. I think also Dr. Coops said before that there are bacteria that they cannot be won by any antibiotic because they are resistant to almost everything. So, of course, it's really crucial that the overuse of one person can lead to resistant strains, and then this will be very bad for the rest of the people. Because this bacteria is not, okay, these are in your gut, but of course, with the feces, they go everywhere in the environment. And you can find antibiotic resistant genes in the wastewater treatments. You can find it everywhere. And these can easily go to the environment and affect all of us.
It's quite interesting to know. And the next question I want to bring up is, what impact do you think that your work does as a freelance researcher have on the world?
Okay, so as a freelance researcher, right now I'm working at the International Hellenic University to the project I just told you about the Helicobacter pylori infection. But also I'm running a small company that we are trying to test the water quality using molecular techniques like NGS techniques and find the microbes that we have in the drinking water. As you all might know, there are several prerequisites all over the world. They are the same, almost the same in Europe or at the US or in Canada. For some bacteria that like enterococcus, E. coli, and some other bacteria that they shouldn't be found in drinking water. And these are specific bacteria that are searched from labs all over the world in order to say that, okay, this water is of good quality, you can drink it. But so far, and what I have searched, there are not any global prerequisites about using molecular techniques, which is very important because there are bacteria that they cannot be found with regular techniques, with cultivation, isolation, and are not Escherichia or other bacteria that are easily cultured in the lab, and they can really degrade the quality of the water we drink. So this is what we are trying to do now here, what I'm trying to do as a freelance since 2016. I'm trying to find, using molecular techniques, all the bacteria that are present in the water, drinking water, and in different steps of drinking water treatment, and also trying to find their numbers using quantitative PCR, and search for specific paths that cannot be detected with regular techniques, and that can potentially degrade water quality. So far, and actually during the last 6 years, this is what I'm mostly working on as a freelancer, apart from the scientific part.
That's really interesting. And now, as our last question, I was wondering, what advice do you have for students that want to either pursue work or research in the field of nutritional sciences?
Okay, so I will kind of copy what Professor Coop said. As you probably understood, I'm working in many fields. Like I'm working with the human gut, with animals' gut, with drinking water, with the rivers. I know that sounds a bit crazy because some people have told me that I'm not focused in something really, like working only with Helicobacter or with a specific disease. But the truth is that I really like what I'm doing. Like I loved it from the first moment I started doing it in 2004. And I'm usually saying yes to all challenges, like, would you like to work on that? I find it very interesting. So, and most of the times I don't feel it, like apart from some bureaucracies that of course I don't like, but most of the times I don't see it as a job because I really like it, I really enjoying, I really being close to nature. I really like reading new things that are being found and trying to work with them. So I would suggest that the best thing to do is try to find something you really like, you really enjoy, and you are not bored with, and try to pursue it.
This is great. Actually, that's something that everyone has to have in mind when pursuing a career in research. So, unfortunately, that's all the time we have for today. So, we'd like to thank you for joining us today. It was a pleasure having you here with us. Now we're going to take some questions from our audience. So, please feel free to leave your questions in the chat below. And after a small break, we'll come back and I'll read them to you.
Okay.
So, see you soon.
Okay.
So we're back, and let's see if there are any questions from the chat. Yeah, sure. So one question for you, Dr. Mazzetti, is from Costas, who's asking if other microbial communities, such as the oral or skin microbiome, can affect the gut microbiome populations.
Yes, there are several studies being performed for that, and in many ways they can do it. The oral or the skin microbiome, they can affect the gut microbiome populations because, or the other way through. And this is, I have not worked so much on this on humans. I mostly know this about animals, that the skin microbiome can really have an effect finally on the gut microbiome, at least for marine animals.
So I think that's all. I don't see any more questions. So, Par?
Yes. So thank you, Dr. Coombs. Thank you, Dr. Mazzetti, once again for joining us today. And I just want to thank all the audience members for tuning in today. We hope you enjoyed this discussion around the gut microbiome, and we hope it inspires you to pursue research in the field of gastroenterology and also in the field of nutritional sciences.
And also, if you're interested to learn more about the work that Synbio is doing and how it's making an impact in the field of gut and digestive health, you can follow us on our Instagram pages and keep up with our work as 2021 iGEM teams. So, thank you.
Thank you and goodbye.
Goodbye.
Thank you. Good luck, guys. Bye.
Thank you.
Bye.
Bye. Bye.